Good evening. Good evening, all of you. Am I audible and visible? If I'm audible and visible, can I see a thumbs up, please? Am I audible and visible? Yeah. So, if you're joining the session for the first time, this is Dr. Shanmugapriya. We are an academy educator for biochemistry. And this session is a high yield MCQ session for FMGE aspirants. So in this session, I've chosen few high yield topics. I will be discussing those topics using few MCQs. Okay, so that's an outline about the session. Before we start the discussion, let me tell you few facts about Unacademy's plans. So because we are celebrating Republic Day, there is a save up to 20% on all medical PG subscriptions, and this is valid till January 31st, 11:59 p.m. So for 36 months subscription, what you would have to pay is just 35,112. And if you're opting for a 12-month subscription, that would cost you 21,183. Okay, so that's for Republic Day. And we also have a target NEET PG 2023 1-month final booster batch, and in this batch there are 3 modules. In the first module, previous year questions will be discussed. In the second module, most expected questions of all 19 subjects will be discussed. And in the third module, there will also be a BTR series by Dr. Zainab. And so NEET PG 2023 All India mock test is on Feb 5th at 9 a.m. And FMG aspirants, there is an iconic subscription plan that is currently at 60% off, so you can try to avail this offer. So that's all about Unacademy's plans. Let's start discussing few MCQs now. So this is the first MCQ. Glucose and fructose are what? Are they functional isomers? Are they optical isomers, stereo isomers, or epimers? Can any one of you try to answer this, please? Functional, optical, stereo, or epimers? Glucose and fructose are functional isomers because glucose is an aldose. Yes, Swaroop, very good. Glucose is an aldose and fructose is a ketose. So I will show you structure of glucose and fructose. The good news is you don't have to memorize any of these structures. Just observe these structures and tell me how many functional groups do you observe in glucose? A functional group that is present in carbohydrate will be either an aldehyde group or a ketone group. In that case, how many functional groups do you observe in glucose? There is only one functional group. Because there is only one functional group, what will you call glucose as? Glucose is a monosaccharide. And what is that functional group which is present? It's an aldehyde group. So it is an aldose. Glucose is a monosaccharide. It is an aldose. How many carbon atoms do you find in glucose? Totally 6 carbon atoms. So glucose is a hexose. Do you understand that? Now look at fructose. In fructose again, how many functional groups do you observe? There is only one functional group. Hi Mota Babi. So in fructose again, there is only one functional group, which means fructose is also a monosaccharide. And what is that functional group which is present in fructose? It is a ketone group. So fructose is a ketose. And how many carbon atoms do you find in fructose? 6 carbon atoms. So it is a hexose again. So what is that you observe between glucose and fructose? Both glucose and fructose are monosaccharides. Both glucose and fructose are hexoses. Both their molecular formula will be C6H12O6. The only difference is glucose has got an aldehyde group as a functional group, whereas fructose has got a ketone group as a functional group. Because their molecular formula is the same and they differ only with respect to the functional group, what will you call them as? You will call them as functional isomers. Got it? You want me to teach electron transport chain? Tomorrow also we have a session, Mota Babi. I will teach you electron transport chain tomorrow. Okay, I can discuss that, but I have not chosen any MCQ based on it. If time permits, towards the end of the session, I'll tell you an outline, but tomorrow we'll discuss electron transport chain completely. Okay, so what are glucose and fructose? Glucose and fructose are functional isomers. What are optical isomers, all of you? Can anybody tell me? Small d and small l, dextrorotatory and levorotatory forms. Yeah, they are optical isomers. So they are based on how they turn the plane polarized light. Dextrorotatory, levorotatory. plus or minus, they are optical isomers. Now what do you mean by stereoisomers? If the molecular formula is the same and the structural formula is also the same, but if they differ only in spatial orientation of their groups, you will call them as stereoisomers. For example, you know this is the structure of glucose, right? We know this is the structure of glucose. In glucose, if you maintain everything, but if you change the orientation of H and OH groups with respect to the second carbon atom alone, if you change the orientation, put OH here and H here, that will be called as mannose. So what are glucose and mannose? They share the same molecular formula, they share even the same structural formula, but they differ only in spatial orientation of their groups. So glucose and mannose are stereoisomers. To be very specific, glucose and mannose are epimers. One type of stereoisomerism is epimerism. Glucose and mannose are epimers. Similarly, glucose and galactose are also epimers, because in the fourth carbon atom, if you change the orientation, if you put OH here and H here, that will be called as galactose. So glucose and galactose are epimers at which position? Fourth position. Okay. So that's about an introduction to isomerism. So if you ask what are the examples of functional isomerism, what will be your answer? Glucose and fructose are functional isomers. What are the examples of epimers? Glucose and galactose are epimers at which position? Second position. Glucose, no, fourth position. Glucose and mannose are epimers at which position? Second position. Okay. Now all of the following are reducing sugars except. Can you answer this question? All of the following are reducing sugars except glucose, fructose, sucrose and maltose. The answer here is sucrose. Okay, sucrose is a non-reducing disaccharide. Do you all understand this? Very good, Swarup. Sucrose is a non-reducing disaccharide. So let me tell you, when will you call it as a reducing sugar? When will you call something as a non-reducing sugar? Any carbohydrate which has got a free carbonyl group will be reducing. And that is why glucose, which has got a free carbonyl group in the first carbon atom, and fructose, which has got a free carbonyl group in the second carbon atom, are both reducing sugars. So when will you call something as reducing sugar? When something has got a free carbonyl group. So glucose and fructose are reducing sugars. But how about sucrose? What is that you know about sucrose? Sucrose is a disaccharide that is made up of glucose and fructose. Okay, sucrose is a disaccharide made up of glucose and fructose. But how do you get sucrose? You get sucrose by linking the first carbon atom of glucose with the second carbon atom of fructose. Whenever you form a disaccharide, one carbon atom of the first sugar should be attached to a carbon atom of the next sugar. So when you get sucrose, it is obtained by linking the first carbon atom of glucose with the second carbon atom of fructose. That way, neither the carbonyl group of glucose nor that of fructose is free. That is why sucrose is non-reducing. So if you ask why is sucrose non-reducing, what will be your answer? Because sucrose lacks a free carbonyl group. And if you asked why does it lack a free carbonyl group, your answer should be because the linkage that is present in sucrose is alpha 1,2. Yeah, because the linkage that is present in sucrose is alpha 1,2. Now do you understand what does 1, 2 mean? It means you get it by linking the first carbon atom of glucose with the second carbon atom of fructose. Okay. Now about reducing and non-reducing sugars. Okay, so if you look at the structure, okay, all monosaccharides are reducing. What gives me the confidence to say that all monosaccharides are reducing? Because every monosaccharide will have either an aldehyde group or a ketone group. In either case, you will find a free carbonyl group. So all monosaccharides are reducing. How about disaccharides? Most of the disaccharides are again reducing. Okay, exception. What are the exceptions? One exception is sucrose. I already told you why is sucrose non-reducing. Because you get sucrose by linking the first carbon atom of glucose with the second carbon atom of fructose. Now apart from sucrose, even trehalose is a non-reducing disaccharide. Why is trehalose a non-reducing disaccharide? Because you get trehalose by linking two glucose residues. How will you get trehalose? By linking two glucose residues. How do they get linked? The first carbon atom of the first glucose with the first carbon atom of the next glucose. That way neither the carbonyl group of the first glucose nor that of the second glucose is free. Do you understand this? So tell me, what are the two non-reducing disaccharides? One is sucrose, the other one is trehalose. Tell me, why is sucrose non-reducing? Because the linkage that is present is alpha 1, 2. Why is trehalose non-reducing? Because the linkage that is present is alpha 1, 1. Okay. So I am going to fill up this tabular column to tell you what are the reducing sugars and what are the non-reducing sugars. Can you tell me what are the reducing sugars, all of you? All monosaccharides are reducing sugars. And among disaccharides, most of the disaccharides are also reducing sugars. What are the exceptions? You know, sucrose is non-reducing and trehalose is non-reducing. And apart from monosaccharides and disaccharides, everything else, all oligosaccharides and polysaccharides. Okay, all oligosaccharides and polysaccharides are non-reducing sugars. Because if you take an oligosaccharide, say a trisaccharide has got 3 carbohydrate residues. And in these 3 carbohydrate residues, all these carbonyl groups will be involved in linkage formation. Only one will be free. So the ratio of the free carbonyl group is to number of sugar residues will be 1:3. For 3 carbohydrate residues, there is only one free carbonyl group. So that is not sufficient enough to bring about the reduction property. So all oligosaccharides and polysaccharides are non-reducing. Okay, so that's about classification of carbohydrates as reducing and non-reducing sugars. Now, try to answer this. All of the following are reducing sugars except. What is the right answer? The right answer here is sucrose. And if you are asking me about maltose, is maltose reducing or non-reducing? What is maltose? Maltose is a disaccharide made up of 2 glucose residues. And what is the linkage that is present in maltose? It is 1,4. So the first carbon atom of the first glucose is attached to the 4th carbon atom of the next glucose. So which carbonyl group is free? The second glucose residue has got a free carbonyl group. So maltose is a reducing disaccharide. So all of you repeat it along with me. What are the non-reducing disaccharides? Non-reducing disaccharides are sucrose and trehalose. Sucrose has got glucose and fructose. Trehalose has got 2 glucose residues. Okay. So the test that is used to detect a reducing substance in the given solution is, what is the right answer? The test that is used to detect a reducing substance in the given solution, all of you please answer. The answer is, very good Swaroop, yes, it is Benedict's test. Okay, so let's try to understand about other tests. What is the use of Biuret test? Biuret test is used to detect proteins. Okay, Biuret test is used to detect proteins. How about Molish test? Molish test is a group test done to detect carbohydrates. Okay, how about benzidine test? Benzidine test is done to detect blood. If you are doing a test to detect the presence of blood in urine, to detect the presence of blood in urine, what is the test we do? It is benzidine test. Okay, so can we learn about Benedict's test now? How do you perform Benedict's test? Please tell me. You take 5 ml of Benedict solution. For performing Benedict's test, we take 5 ml of Benedict solution, to which you add 8 drops of the solution to be tested, and then we heat it. And suppose there is a reducing substance in the given solution, you will observe a color change from blue to a series of colors. From blue, initially it becomes green, then it becomes yellow, orange or red. And the color will depend upon the concentration of the reducing substance that is present in the given solution. Don't we know this? So how do you perform Benedict's test? You take 5 ml of Benedict solution, you add 8 drops of the solution to be tested and then be heated. Suppose there is a reducing substance in the given solution, there will be a color change from blue to a series of colors. It can become green or yellow or orange or red. That will depend upon the concentration of the reducing substance that is present in the given solution. Now can you tell me what is the composition of Benedict's reagent? What is the composition of Benedict's reagent? I have already given you a clue about the first component of Benedict's solution. I said it is blue in color. So what gives the blue color? It is copper sulfate. So what is the first component of Benedict's solution? It is copper sulfate. And about the second component, any reducing substance will exhibit its reducing property effectively in an alkaline medium. Yeah, any reducing substance will exhibit its reducing property effectively in an alkaline medium. So the second one is sodium carbonate, which provides a required alkaline medium. And the third one is sodium citrate, which stabilizes the solution. So what is the composition of Benedict's solution? Copper sulfate, which provides a blue color, sodium carbonate, which provides an alkaline medium, and sodium citrate, which stabilizes the solution. And this is a test that is done to detect the presence of any reducing substance in the given solution. Okay. So try to answer this. Pyrimidine ring is derived from what? Derived from all except. Pyrimidine ring is derived from all except carbon dioxide, glutamine, aspartate and glycine. The answer is glycine. Glycine does not act as a source of pyrimidine ring, it acts as a source of purine ring. Do you understand this? Glycine acts as a source of only purine ring. And before we start discussing what is the source of which atom of pyrimidine ring, I want you to know few facts about the ring. Yeah, pyrimidine ring, when you compare it with purine and purine has got a smaller name, but it has got a bigger structure. Purine has got 2 rings and 9 atoms. Whereas pyrimidine with a longer name has got a smaller structure. It has got single ring and 6 atoms. So tell me how many atoms are there in a pyrimidine ring? There are 6 atoms. Now what are the sources of each and every atom of these 6 atoms? Okay. For this, I think you must have learnt about urea cycle and pyrimidine synthesis. Both urea cycle and pyrimidine synthesis, the first step is catalyzed by the same enzyme. Which enzyme is it? It is carbamyl phosphate synthetase. Please memorize it. Contribution from aspartate, glutamine and carbon dioxide. Good evening, Ashwin Yadav. Yes, you're right. So what is the first step in urea cycle and pyrimidine synthesis? In both the cases, the first step is catalyzed by carbamyl phosphate synthetase. The urea cycle enzyme is named as carbamyl phosphate synthetase 1. Pyrimidine synthesis enzyme is named as carbamyl phosphate synthetase 2. Okay. The next difference between urea cycle and pyrimidine synthesis is carbamyl phosphate synthetase 1 is present in mitochondria. Carbamyl phosphate synthetase 2 is present in cytoplasm. The third difference is after carbamyl phosphate is formed, the next enzyme in urea cycle is ornithine transcarbamylase. What is the next enzyme in urea cycle? It is ornithine transcarbamylase. Whereas in pyrimidine synthesis, it is aspartyl transcarbamylase. So tell me what are the 2 enzymes which are involved in pyrimidine synthesis? It is carbamyl phosphate synthetase 2 and aspartate transcarbamylase. Okay. So if you know that the first enzyme is carbamyl phosphate synthetase 2, there should be a carbon donor. Is it not true? There should be a carbon donor. What will be the carbon donor? It is carbon dioxide. So the first substrate is carbon dioxide, carbamyl. You need an amino group donor and that is glutamine. Phosphate donor is ATP. So in the first step, carbon dioxide reacts with glutamine and ATP to form carbamyl phosphate. In this case, carbon dioxide acts as a source of C2. You write carbon dioxide as CO2, which acts as a source of C2. Glutamine will act as a source of N3. Please memorize. In the case of both purine ring and pyrimidine ring, N3 is from glutamine. Both purine and pyrimidine, N3 is from glutamine. So now you know the source of C2 of pyrimidine ring, you know the source of N3 of pyrimidine ring. So what will be the source of the remaining 4 atoms? I said the next enzyme is aspartyl transcarbamylase. So the remaining 4 atoms are from aspartic acid. So what are the remaining 4 atoms? N1. Okay, N1, C4, C5, C6. N1, C4, C5 and C6 are all from aspartic acid. Is it not simple now? So tell me, for pyrimidine synthesis, how many atoms do you find in pyrimidine ring? There are 6 atoms. What are the sources of these 6 atoms? I said N1 is from aspartic acid. Right? N1 is from aspartic acid. C2 is from carbon dioxide. And then N3 is from glutamine. Please memorize it. N3 of purine and pyrimidine are from glutamine. And the remaining atoms are from aspartic acid. Okay. Here, what is the rate limiting enzyme of pyrimidine synthesis? For pyrimidine synthesis, the rate limiting enzyme is carbamyl phosphate synthetase 2. Please memorize all these facts. Yeah, the rate limiting enzyme of pyrimidine synthesis is carbamyl phosphate synthetase 2. So what do you think is the rate limiting enzyme of purine synthesis de novo? For purine synthesis de novo, the rate limiting enzyme is glutamine PRPP amidotransferase. So what is the rate limiting enzyme of purine synthesis de novo? It is PRPP glutamine amidotransferase. Okay. So now I am going to tell you about the sources of all the atoms of a purine ring. What did I tell you about purine ring? I said it has got 2 rings and 9 atoms. So what is the source of N1? What did I tell you is the source of N1 in pyrimidine ring? Aspartic acid. To remember, first alphabet is A, right? First alphabet is A. So N1 of both purine and pyrimidine are from aspartic acid. Similarly, N3 of both purine and pyrimidine are from glutamine. Not only N3, even N9 is from glutamine in purine ring. Okay. Now C2 and C8. C2 and C8 are from tetrahydrofolate derivatives. If you are asking me how to memorize it, C2, 8, right? 2 into 4 is 8. True or false? True. In 2 into 4 is 8. 4 is tetra. So 2 and 8 are donated by tetrahydrofolate derivatives. Okay. And then C6 is from carbon dioxide. Okay. C2 of pyrimidine ring is from carbon dioxide. C6 of purine ring is from carbon dioxide. And apart from this, what is left out? C4, C5 and N7. Okay. C4, C5 and N7 together will be from glycine. C4, C5 and N7 together will be from glycine. So tell me, what are the atoms of purine ring I have told you about? Yeah, how many atoms are there in a purine ring? Purine ring has got 9 atoms. So what are the sources of all these 9 atoms of purine ring? As I told you, the first one. What is the first one? N1. N1 is from what? The first alphabet is A. It is aspartic acid. N1 is from aspartic acid. N3 and N9 are from what? Both N3 and N9 are from glutamine. C2 and C8. 2 into 4 is 8. That will be from tetrahydrofolate derivatives. Okay. C6 is from carbon dioxide. Apart from this, C4, C5 and N7, all these are from glycine. Which is the rate limiting? Can the light chat be made active, please? Which is the rate limiting enzyme of pyrimidine synthesis? For pyrimidine synthesis, it is aspartyl transcarbamylase. For pyrimidine synthesis, it is carbamyl phosphate synthetase 2. For purine synthesis de novo, it is glutamine PRPP amidotransferase. Okay. So what is the right answer here? Pyrimidine ring, N1 is from aspartate, right? N1 is from aspartate, and then you have carbon dioxide. But what is not a source? Yeah, glycine is not a source. Okay. Answer this please. DNA is extracted from a blood sample and is subjected to dehydration. The form of DNA that is expected is After dehydration, what is the form of DNA that you get? Do you all know that the most common physiological form of DNA is B type? The most common physiological form of DNA is B type. On dehydration, B type becomes A. Yeah, B type becomes A. So the answer is choice B. So let's try to understand. It's not cDNA, Swarup. On dehydration, it is A DNA. What is cDNA? cDNA is complementary DNA. What is it complementary to? It is complementary to mRNAs. cDNAs or complementary DNAs are complementary to fully spliced mRNAs. Okay? And they will be complementary DNA. Again, physiological form is B type. So on dehydration, it takes up A type. So let's try to understand about DNA structure. So first about Watson and Crick model. What did Watson and Crick state? According to Watson and Crick, the two strands are exactly complementary and anti-parallel. Don't we know that? If there is A on one strand, there should be T on the other strand. If there is G on one strand, there should be C on the other strand. Good evening, Shweb. So they're exactly complementary. What do you mean by being anti-parallel? If one strand is 5 prime, 3 prime, the other strand will be 3 prime, 5 prime. And the two strands are oriented in a ladder-like fashion. Don't you think that it looks like a ladder? The backbone of the ladder, what I'm marking as a black strand here, the backbone of the ladder is made up of phosphodiester linkages. Yeah, they're made up of phosphodiester linkages with ribose or deoxyribose sugars. Can you see that here? This is the backbone. What is the backbone made up of? 3 prime, 5 prime phosphodiester linkages and ribose or deoxyribose sugars. The steps are made up of nitrogenous bases which are linked by hydrogen bonds. There are 2 hydrogen bonds between A and T and 3 hydrogen bonds between G and C. So can you tell me what is the linkage which stabilizes a double-stranded DNA? A double-stranded DNA is stabilized by hydrogen bonding. Okay, so this is all about Watson and Crick model of DNA. So what did Watson and Crick state? According to Watson and Crick, the two strands are exactly complementary and anti-parallel. They are oriented in a ladder-like fashion. The backbone of the ladder is made up of phosphodiester linkages with ribose or deoxyribose sugars. Steps are made up of nitrogenous bases linked by hydrogen bonds. 2 hydrogen bonds between A and T and 3 hydrogen bonds between G and C. And based on this structure, based on Watson and Crick's DNA model, there are 7 conformations which are possible. Out of these 7 conformations, the most common physiological form of DNA is B type. Okay, so what is the special feature of B type? It is the most common physiological form of DNA. And it is a right-handed helix. This is often asked. If they ask you which is the left-handed helix, what will be your answer? Yeah, left-handed helix. Left-handed helix is Z type. Which is the only which is the only left-handed helix? It is Z type. So B type is a right-handed helix. And in B type, for every full turn, there are 10 base pairing. What do you mean by full turn? From here till here. In B type, for every full turn, there are 10 base pairing. And every base pairing rises the height by 3.4 angstrom. So one full turn with 10 base pairing will measure 34 angstrom. And the width will be 20. Okay, and the width will be 20. So, and for every full turn, there is always a major groove. Do you see that this groove is deeper? So, for every full turn, there is always a major groove and a minor groove. So, that's all about B type. So, tell me what are the facts that I told you about B type? It is the most common physiological form of DNA. And B type is a right-handed helix. Here for every full turn, how many base pairing? 10 base pairing. Every base pairing rises the height by 3.4 angstrom. One full turn with 10 base pairing measures 34 and the width is 20. And for every full turn, there is a major and a minor groove. Okay. The next common conformation is A type. When will you find A type? Whenever DNA undergoes dehydration. Okay, whenever DNA undergoes dehydration, or whenever there is a DNA-RNA hybrid, or whenever there is RNA-RNA hybrid. If it's not a completely double-stranded DNA, then it takes up A type. So when do you find A type? DNA on dehydration, or whenever there is a hybrid between DNA and RNA or RNA-RNA hybrid, it is A type. And this A type is also a right-handedly coiled helix. This is also right-handedly coiled. And in A type, for every full turn, you find 11 base pairing. And in A type, you don't find major or minor grooves. All grooves are of the same dimension. So tell me, what are the facts that I told you about A type? A type is found whenever DNA undergoes dehydration, or whenever there is a hybrid between DNA and RNA or RNA-RNA hybrid. It is also a right-handedly coiled helix. Here there are 11 base pairing. No major or minor groove. All grooves will be of the same dimension. Okay. The next commonly asked conformation is Z type. Okay. And this Z type is found in those regions of your chromosome which are rich in GC sequences. So can anybody tell me what is chromosome? The chromosome which you draw with a centromere, a short arm and a long arm, is nothing but one long double-stranded DNA which has got condensed with the help of proteins. Do you all understand this? So what is one chromosome? One chromosome is nothing but one long double-stranded DNA which has got condensed with the help of proteins. So in this double-stranded DNA, most of the regions will have equal number of AT sequences and equal number of GC sequences. But some regions will have more GC sequences. So those parts of your chromosome which have more GC sequences will take up what conformation? Z type of conformation. Okay, and this is the only left-handedly coiled conformation. And in Z type, for every full turn you find 12 base pairing. So what are the facts that I told you about Z type? You find Z type in those parts of your chromosome which are rich in GC sequences. You find 12 base pairing here. It is a left-handedly coiled conformation. So having known this, tell me the answer. DNA is extracted from a blood sample and is subjected to dehydration. What is the form of DNA that you observe, all of you? On dehydration, it is A type. Okay. Blood glucose is maintained by all of the following except. What do you think is the right answer? Is blood glucose maintained by dietary glucose, muscle glycogenolysis, liver glycogenolysis and gluconeogenesis? What is the right answer, all of you? Schwab says it's dietary glucose. Is that true, Schwab? How do you maintain plasma glucose, all of you? Why should you maintain plasma glucose? First tell me the answer. Why do you have to maintain plasma glucose? It is because neurons, principal cells of your body, namely neurons and RBCs, are dependent only on glucose. So at any point in your life, you should keep maintaining plasma glucose. And how do we maintain plasma glucose? We have three ways. The first way is dietary glucose. Obviously, right? Any dietary carbohydrate that you take gets digested, gets absorbed as glucose. That will maintain plasma glucose. And this dietary glucose can maintain plasma glucose only till 2 to 2.5 hours after the previous meal. Suppose you have your lunch. Yeah, you have your lunch at 1 p.m. Till 3 to 3:30 p.m. your dietary glucose will maintain plasma glucose. After which contribution from dietary glucose decreases. But you cannot allow your plasma glucose to decline. So how do you maintain plasma glucose? That is done by glycogenolysis. So the second way by which you maintain plasma glucose is glycogenolysis, and finally it is gluconeogenesis. After glycogenolysis, it is gluconeogenesis. And as far as glycogenolysis is concerned, I am going to tell you that glycogenolysis can happen in, what is that about muscle glycogenolysis, Varu? Glycogenolysis can happen in liver and in muscle. Don't you all know that glycogen is stored in liver and muscle? So both liver and muscle can undergo glycogenolysis. But only liver glycogenolysis can maintain plasma glucose, muscle glycogenolysis cannot. Why do you think muscle glycogenolysis cannot? It is because glycogenolysis is done by an enzyme called as glycogen phosphorylase. So if you are asked what is the rate limiting enzyme of glycogenolysis, what will be your answer? The rate limiting enzyme of glycogenolysis is glycogen phosphorylase. And as the name indicates, this enzyme adds inorganic phosphate and then it cleaves glycogen. When it cleaves glycogen, one glucose is removed from glycogen and that glucose gets attached to the phosphate that is used for cleaving. When glucose gets attached to phosphate, the product which you get is glucose 1-phosphate. So all of you repeat it along with me. Yeah, repeat it along with me. What is the product of glycogenolysis? Product of glycogenolysis is not free glucose, it is glucose 1-phosphate. Now this glucose 1-phosphate will get converted to glucose 6-phosphate in the presence of mutase. Okay, glucose 6-phosphate is formed, that happens in the presence of mutase. And all these are happening within the cell, within hepatocyte or within the muscle cell. And for this glucose 6-phosphate to reach the circulation, it should become glucose. How will you be able to convert glucose 6-phosphate into glucose? With the help of glucose 6-phosphatase. And this glucose 6-phosphatase is present only in liver. So only liver glycogenolysis can increase blood glucose, muscle glycogenolysis cannot. Because a product of muscle glycogenolysis is just glucose 6-phosphate which cannot reach the circulation. Is that clear, all of you? Yeah. So what have you learned? I told you 2 facts now. The first fact is the rate limiting enzyme of glycogenolysis is glycogen phosphorylase. The second fact is product of glycogenolysis is not free glucose, it is glucose 1-phosphate. The third fact that I told you is only liver glycogenolysis can increase blood glucose, muscle glycogenolysis cannot, because muscle lacks glucose 6-phosphatase. Okay. So tell me, what are the pathways which can maintain blood glucose? One is dietary glucose. It can maintain blood glucose till 2 to 2.5 hours after the previous meal. And then it is liver glycogenolysis. Liver glycogenolysis starts 2 to 2.5 hours after the previous meal, and it can maintain blood glucose till 12 to 18 hours of starvation. Liver glycogenolysis can maintain blood glucose till 12 to 18 hours of starvation, after which it has to be gluconeogenesis. And gluconeogenesis begins 6 hours after the previous meal. Okay, though glycogenolysis is also continuing, gluconeogenesis begins 6 hours after the previous meal and can go on as long as it's possible. As long as the person is alive, it can go ahead. Okay, so that's about what are the pathways which can maintain blood glucose. So what is the right answer here? It is maintained by all of the following except. It can be maintained not just till 24 to 48 hours. Yeah, it can go on even for few days. Gluconeogenesis, okay, till 5 to 7 days. So what is the right answer? The right answer is muscle glycogenolysis can never increase blood glucose. Very good, Ghost Rider. Now tell me the answer. Glucagon stimulates what? Glucagon stimulates muscle glycogenolysis. It stimulates liver glycogenolysis. Both none. Can anybody answer? Shweb, Ghost Rider, Swarup, Acharya, N. Anyone else? Ashwin? C. Okay. Shweb says it's C. Do you think? Very good, Swarup. Swarup is right. Glucagon stimulates only liver glycogenolysis. Glucagon cannot stimulate muscle glycogenolysis, because what is the only aim of glucagon in your body? Though glucagon has got receptors in almost every target tissue that is possible, working together, glucagon has got only one aim, that is to increase plasma glucose. So glucagon will stimulate only those pathways which can increase plasma glucose, because glucagon knows that muscle glycogenolysis cannot increase plasma glucose. It does not touch it. Do you understand this? So glucagon stimulates what? Only liver glycogenolysis. It does not stimulate muscle glycogenolysis. What about this? Epinephrine stimulates what? Shweb, Swarup, one of you answer. Epinephrine stimulates what? Epinephrine stimulates both. Why should epinephrine stimulate both? Because epinephrine is a stress hormone. Yes or no? Ghost Rider. Epinephrine is a stress hormone. What are you supposed to do when you are stressed? Yeah, I'll give you a hypothetical scenario. Yeah, what will you do when you are chased by a dog? If you are chased by a dog, what will you do first? You should start running. And for you to run, what should be active? Your muscles should be active. And for your muscle to be active, muscle glycogenolysis should be stimulated. Right? But can you run for a long time? Yeah, can you chase against a dog for a long time? No. So you should be smart enough to go and close a gate behind you. For you to be smart enough, what should be active? Your brain has to be active. And for your brain to be active, you should increase plasma glucose. How will you be able to increase plasma glucose? By stimulating liver glycogenolysis. So epinephrine stimulates both muscle glycogenolysis and liver glycogenolysis. Do you understand this? This is what you are supposed to do whenever you are stressed. You should first run away from the stress and you should be smart enough to overcome the stress. In either case, epinephrine stimulates both liver glycogenolysis and muscle glycogenolysis. Okay. All of the following are gluconeogenic substrates except. Tell me the answer. Glycerol, lactate, alanine, acetyl-CoA. All the following are gluconeogenic substrates except. Anybody? The right answer here is acetyl-CoA. Acetyl-CoA is not gluconeogenic. Very good, Swaroop. Acetyl-CoA is not gluconeogenic. So when will you call something as gluconeogenic? Think and tell me. Do you know that gluconeogenesis is reversal of glycolysis, all of you? Do you all know that gluconeogenesis is reversal of glycolysis? Because in glycolysis, you take one glucose and you split it into two molecules of pyruvate. So what will you do in gluconeogenesis? You start with two molecules of pyruvate, you end up in one molecule of glucose. Okay, so you are going to just reverse glycolysis. So when will you call something as a gluconeogenic substrate? Only when a substance on catabolism can give rise to one of the glycolytic intermediates or one of the citric acid cycle intermediates other than acetyl-CoA, you can call them as gluconeogenic. Why? Because if they can give rise to any of the glycolytic intermediates, whatever may be the intermediate it gives rise to, from that point you can reverse glycolysis, you can reach glucose. Right? What if it gives rise to citric acid cycle intermediates? Whatever may be the intermediate gives rise to, at the end of citric acid cycle, you regenerate oxaloacetate. Don't you all know that? Even if you don't know that till today, please memorize it. In citric acid cycle, acetyl-CoA condenses with oxaloacetate and then finally you regenerate oxaloacetate. So oxaloacetate is considered as a catalyst of citric acid cycle. In every cycle you use oxaloacetate and you regenerate oxaloacetate. So if a substance on catabolism gives rise to one of the citric acid cycle intermediates, at the end of citric acid cycle you will regenerate oxaloacetate. And this oxaloacetate is a gluconeogenic substrate. It can start getting into gluconeogenesis, can reach glucose. So tell me, what is the criteria that you should have in your mind? Yeah. What is the criteria that you should have in your mind for you to call something as gluconeogenic? Only when a substance on catabolism gives rise to a glycolytic intermediate or a citric acid cycle intermediate other than acetyl-CoA, you can call them as gluconeogenic. What if it gives rise to acetyl-CoA? Acetyl-CoA cannot get into gluconeogenesis because pyruvate dehydrogenase complex, which converts pyruvate to acetyl-CoA, is an irreversible step. Pyruvate dehydrogenase complex catalyzes an irreversible step. So from pyruvate you can form acetyl-CoA, but from acetyl-CoA you can never form pyruvate. Do you understand this? From acetyl-CoA you can never form pyruvate. If you cannot form pyruvate, it cannot get into gluconeogenesis. Okay, so acetyl-CoA is never gluconeogenic. That is why even chain fatty acids, please all of you memorize it. All even chain fatty acids are never gluconeogenic. They cannot get into gluconeogenesis because all even chain fatty acids on catabolism will give rise to acetyl-CoA, and acetyl-CoA is never gluconeogenic. And if you are asked what are the principal gluconeogenic substrates, what will be your answer? Glycerol, lactate, alanine. Yeah, please memorize it. What are the three gluconeogenic substrates? Glycerol, lactate and alanine. Okay. Now try to answer this. A child presents with hypoglycemia, hypophosphatemia, jaundice and hepatomegaly after transitioning from mother's milk to infant foods. The probable enzyme defect is. Tell me the answer. Can we start excluding all the choices one by one? Yeah. So can it be lactase deficiency? Can it be lactase deficiency? What do you call lactase deficiency as? It is called as lactose intolerance. Don't you know that? Lactase deficiency is nothing but lactose intolerance. So tell me, what is lactose intolerance? It's caused by a defect of lactase in the intestinal villi. Up. What is it? I am not riding. No, I am at home. So what is lactose intolerance? Lactose intolerance is caused by the defect of lactase in the intestinal villi. So when this enzyme is defective, any lactose that is present in the diet, any lactose that is present in the diet cannot be digested. If it cannot be digested, it cannot be converted to galactose and glucose. As long as lactose stays as a disaccharide, it cannot be absorbed along the intestine. Only when something becomes a monosaccharide, it can be absorbed into the circulation. Because here there is lactase deficiency, lactose is not digested to give glucose and galactose. So lactose stays back as a disaccharide in the intestinal lumen. Because lactose is osmotically active, it attracts water. And this water causes osmotic diarrhea. Do you understand this? Water causes osmotic diarrhea. So tell me, what is one manifestation of lactose intolerance? It is osmotic diarrhea. Okay. And when lactose is not getting digested by an intestinal enzyme, undigested lactose will reach the colon. And in colon, microorganisms start attacking this lactose and they convert lactose into methane and hydrogen. All colonic microorganisms attack lactose in the colon and they convert lactose into hydrogen and methane. Both hydrogen and methane are gases. Because in this case gases accumulate in the colon, the child will present with bloating. The child will present with bloating, flatulence and frothy stools. So tell me, what is the reason for lactose what is the reason for bloating, flatulence and frothy stools and lactose intolerance? It is because lactose is not digested in the small intestine. Undigested lactose reaches the colon, and in colon it's converted to hydrogen and methane, both of which are gases, and gases are responsible for bloating, flatulence and frothy stools. Sometimes colonic microorganisms will convert this lactose into an acid, and that is the reason for acidic pH of stools. Okay, and only because stool's pH is acidic, the child will present with perianal excoriation. It will present with perianal excoriation. I think this was asked in the latest FMGE exam, right? FMGE, recall, we had the discussion. The question was, breastfeeding is contraindicated in which of the following conditions? Breastfeeding is contraindicated in lactose intolerance and in galactosemia. Why is it contraindicated in lactose intolerance? Because when lactase is absent, the lactose that is present in the breast milk will not be digested. That is going to cause osmotic diarrhea, bloating, flatulence, frothy stools and acidic pH of the stools. Why is it contraindicated in galactosemia? Following intake of milk, lactose gets digested to give glucose and galactose. Galactose will enter into the circulation, and galactosemia is a galactose metabolism disorder that will present with all complications. Do you all understand this? Okay. So that's all about lactose intolerance. So do you understand that lactose intolerance is a digestion defect? Yes or no? It's not a metabolic defect, it's a digestion defect which is happening in the intestinal lumen. So it will present only with gastrointestinal disturbances like diarrhea. It is not going to present with metabolic manifestations like hypoglycemia. Whereas what is given in the history? In the history, the child, what is your doubt, Ghost Rider? What is your doubt about galactosemia? Galactosemia, you are asking me for a cause. Galactosemia can be caused by either galactokinase deficiency or it can be caused by galput deficiency. What is galput? It is galactose 1-phosphate uridyl transferase. Do you understand that? Galactosemia is a metabolic defect which is happening within the cell after galactose gets absorbed. Okay, it is caused either by galactokinase deficiency or by galactose 1-phosphate uridyl transferase deficiency. Okay, so a child presents with hypoglycemia, hypophosphatemia, jaundice, hepatomegaly after transitioning from mother's milk to infant foods. Okay, so do you understand that this cannot be galactosemia? Had it been galactosemia, the child should have presented with all these features even when the child was mother fed. Because you know, mother's milk is lactose, lactose gives rise to galactose, and all these features should have appeared even then. So I am excluding galactosemia, which is galactose 1-phosphate uridyl transferase defect. Can it be aldolase A defect? Aldolase A is an enzyme of glycolysis. If glycolysis is defective, it does not present with hypoglycemia. It will present with hemolytic anemia. Do you understand this? Whenever there is a glycolytic enzyme defect, what will it present as? It will present as hemolytic anemia because RBCs are completely dependent on glycolysis. Do you all know that? Yeah, RBCs are completely dependent on glycolysis because RBCs do not have mitochondria. Without mitochondria, they are dependent only on anaerobic glycolysis. In that case, whenever there is any glycolytic enzyme defect, which cell gets affected the most? RBCs will get affected the most. So that will present only as hemolytic anemia. So having excluded all these three choices, what are you left with? We are left with only one choice, which is aldolase B. Aldolase B defect is called as hereditary fructose intolerance. And only when there is hereditary fructose intolerance, classically the history will be like until the child is mother fed, there will be no manifestations. After transition from mother's milk to infant foods, because every infant food is loaded with sucrose. Yeah, because manufacturers of all these infant foods will want the infant to love it. So what do they add? They add table sugar. What is table sugar? Sucrose. Sucrose will get digested to give glucose and fructose. And fructose will start getting into the system and then it presents with manifestation that is called as hereditary fructose intolerance caused by aldolase B defect. Okay, so I am going to summarize few facts now for your benefit. So what are the facts that I want you to recollect? I want you to know about few disaccharides. What are the disaccharides I told you about? First disaccharide I told you is sucrose. What is sucrose made up of? Glucose and fructose. And why is sucrose non-reducing? Because the linkage that is present in sucrose is alpha 1,2. And the second disaccharide that I told you about is trehalose. Trehalose is made up of glucose and glucose. This is also a non-reducing disaccharide because the linkage that is present is alpha 1,1. Okay, and what are the other disaccharides I told you about? Lactose. What is lactose made up of? Lactose, which is present in milk, is a disaccharide. It is made up of galactose and glucose. It's made up of galactose and glucose. Okay, and then I told you table sugar is sucrose, glucose and fructose. Okay, so if you know about these disaccharides, that is enough. And what did I tell you? Lactose intolerance is caused by what? Lactose intolerance is caused by lactase deficiency in the intestinal villi. What will it present as? It will present only as GI disturbances like diarrhea. Okay. Whereas fructose intolerance. Fructose intolerance is a metabolic defect caused by aldolase B defect. And then galactosemia is caused by the defect of what? Classical galactosemia is caused by the defect of galput. What is galput? Galactose 1-phosphate uridyl transferase. Okay. So that's all for today. Okay. So I'll meet you soon with another set of MCQs, another set of high yield MCQs using which I'll help you understand probably lipid metabolism and protein metabolism. Okay. So thank you. Have a good night.